So our guest speaker is Jana Pachlopnik Schmid, who is, I think, known to many of us, has been interested in HLH for a number of years and has been a member. And so Jana will be, sorry, Jana's background, I should say, I didn't realize this, but is entirely Swiss, right? So she got her degree, MD and PhD in Bern. She did then went and did a postdoc with Alain Fischer and Geneviève de Saint-Marcel and at the Neckar in Paris. But then she returned back to Switzerland, to Zurich, where she is now a professor and the head of her division. She's focused on immune regulatory and other immune deficiency disorders, obviously with a special interest in HLH and in translational therapeutics. So Jana will be speaking to us today about new findings relevant to HLH. Now, do we have, Kathy, I should ask, do we have a housekeeping slide before I turn this over to Jana? Yep, so just remind everybody, keep yourself muted during the presentation. If you have a question, use the raise hand function. At the end of Jana's talk, we can then get to your question and you'll be called on, at which point you can unmute yourself and ask your question. So, but at this point, I'm going to hand it over to Jana, who will then, I think you should be able to control the slides from your desktop. We see you, and if you want to share your screen, then we should be able to see your slides as well.
Can you see my slides, Michael?
Yes. Yep. I can see it. You just want to go into presentation mode, and I can hear you fine as well. Great. Thank you.
Thank you very much, Michael. Good morning. Thank you for the nice introduction, and good morning, dear colleagues. It's a real pleasure to give a talk about hemophagocytic lymphohistiocytosis at this year's Histiocyte Society meeting. And thank you very much, Michael, for the invitation. It is really a great honor to do this. Thank you. So these are my disclosures. And as Michael already told you, with my research team, we are especially interested in inborn errors of the immune system and what these diseases can teach us about the functioning of the immune system. So a simple view of what is happening when the immune system is functioning normally is that the body fights against viruses, fungi, parasites, and that it gets rid of the body's own infected cells. But a moment of reflection shows that this cannot be the whole story. We are exposed to so many germs and every day, and our immune system must not act to everything. More subtly, our immune system must be able to tell the difference between harmless and maybe even useful bacteria, such as those in our gut or on our skin, and which should be left alone, and dangerous bacteria or other germs, or even our own malignant cells, which we generate every day, and that could make us sick, which had to be dealt with. And even though it is true that the immune system targets what might become dangerous, it has become apparent that multiple biological checkpoints and balances run by countless cells and molecules regulate this process. If this immune system is completely turned off, we will be susceptible for infections and also for cancer. But on the other hand, if the immune system gets overactivated and cannot stop ongoing activation, healthy cells and tissues will be destroyed. So let me illustrate this with a case report. A previously healthy 6-year-old boy presented after a history of 10 days of fever, and 5 days earlier, he was diagnosed with SARS-CoV-2 infections by RT-PCR from a nasopharyngeal swab. On admission, physical examination was remarkable for fever, tachycardia, hypertension, and respiratory distress, and the boy was suffering from abdominal pain. Empiric antibiotic treatment was started, and the boy was transferred to the intensive care due to ARDS. As the patient fulfilled the diagnostic criteria of MISC, or multisystem inflammatory syndrome related to COVID-19 at the time of admission, treatment with high-dose intravenous immunoglobulin was started, steroids, and as the boy's general condition deteriorated the next 24 hours, IL-1 receptor blocker anakinra was added. The patient's clinical condition continued to worsen, and he developed liver failure, profound coagulopathy, and highly suggestive of HLH. And the same day, the results of a PCR from blood came back showing very high EBV copy numbers, and therefore the IL-1 blocker was stopped, and HLH-directed treatment was started. Despite maximal treatment and liver support using a molecular absorbent recirculation system, the MARS, the patient's conditions severely deteriorated, and he succumbed on day 8 after admission. And we included this patient post-mortem in our genetic study of immunodeficiencies and found a deleterious mutation in the SAP gene, and therefore made the diagnosis of XLP1. Thus, our patient presented with fever and rapidly evolving ARDS, and due to previous infection with SARS-CoV-2, the suspected initial diagnosis was MISC. He ultimately died secondary to liver failure as a consequence of EBV-triggered HLH in the context of XLP1. So these two disease entities, MISC and HLH, have an overlapping clinical spectrum and also lab values. But if you compare our patients' laboratory values with the ones of a total of 58 patients with MISC described by Lisa Whittaker and colleagues, you will see that there are some key distinguishing findings. Our patient had, compared to MISC, lowish neutrophils, normal lymphocyte number, as opposed to lymphopenia in MISC, lowish CRP, high ferritin than the MISC patients, and lower fibrinogen. So what the clinical history of this patient sadly exemplifies is, first, how difficult it can be to recognize this one single patient with a rare disease whose clinical presentation and lab values are, at least at the beginning of the disease, only slightly different from other patients with more frequent diseases. And second, it exemplifies that patients with inborn errors of immunity who lack one single component of the immune system, namely the adaptive protein SAP in the case of the boy, can teach us a lot about the importance of a certain gene product for the functioning of the whole system. And we can learn from these patients concerning, we can learn a lot from these infection during defense against malignant diseases, and we can also learn which components of the immune system are needed to calm down the reaction to regulate the immune system. And one of the most fascinating, but clinically probably also most challenging immunodeficiencies with immune dysregulation is the group of syndromes with hemophagocytic lymphohistiocytosis. Natural mutants that affect the cytotoxic machinery of lymphocytes provide evidence of a critical role played by the cytotoxic pathway in the regulation of immune homeostasis. Patients with germline defects in this pathway, so defects in perforin or in one of the components important for the transport, docking, and degranulation of the perforin-containing granule at the plasma membrane towards the target will almost inevitably suffer from HLH. But why does a defect in lymphocytic cytotoxicity lead to this terrible cytokine storm syndrome? In a healthy immune system, cells which are infected with an intracellular pathogen will be killed by CD8 T cells or NK cells. These lymphocytes, in the case of deficient cytotoxicity, are still able to recognize the infected cell. They are still able to secrete cytokines and to proliferate, and they will do so permanently in a vicious circle because the trigger is not removed. In 2010, at least, this was generally assumed, although there was no formal proof for it. And this came later, and then I will summarize some of these publications which show experimental evidence and help to understand the pathogenesis of HLH. So this very nice study by Jenkins and colleagues shows that the synapse time between the cytotoxic lymphocyte and the target cell is increased if the cytotoxic machinery is deficient, while PI, so propydium iodide, shown here in red, as a sign of cell permeabilization, so here made permeable by perforin, increase in target cells within minutes after encountering a perforin-sufficient lymphocyte. This will not happen in perforin deficiency. So this was already known before. But what was new and what was shown here by Jenkins and colleagues was that in contrast to perforin sufficient lymphocytes, the perforin deficient ones had a much higher and longer calcium flux shown and increased cytokine production, as shown here. As shown in the lower right corner. And this was
here we go. Yeah, so we lost you for a bit.
I lost you. Yeah. I'm really sorry. I don't know why this happened, actually. So I'm just going to continue.
Yes, you can pick up. Things went black for a bit there. I don't know how long you noticed you were out for. But can you share your screen again?
Yes, I'll do so. Can you see it now?
Yeah, we see it fine. We hear you fine.
Oh, okay. So the second paper by showed that the fratricide killing is another important pathomechanistic aspect of the pathogenesis of HLH. And what they have shown is when they blocked NK cells by NKG2D blocking antibody, which will block NK cells, they observed much higher numbers of CD8 T cells in mice that had been infected with lymphocytic choriomeningitis virus, or LCMV, which is a model for HLH. And another paper by Wagoner and colleagues, they showed that the mice infected with another virus had a better viral clearance after NK cell depletion, but these mice then died due to immunopathology. Because these mice did have more virus-specific CD4, the authors claimed that NK cells cytolytically eliminate CD4 cells, which affect the CD8 T cells. So taken together, these papers show that show a regulatory function of NK cells, so either directly via killing of CD8 T cells or via killing CD4 T cells. Besides NK cells, also regulatory T cells might play a role in the pathogenesis of HLH. And the research group of Adrian Liston has shown that the proportion of regulatory T cells, as you can see on the left-hand, on the figure on the left-hand side, the proportion of regulatory T cells is decreased in patients and also in mice during HLH. The authors showed that the Treg population was decreased due to a consumption of IL-2 by activated CD8 T cells. In vivo administration of IL-2 complexes could restore the regulatory T cell pool. So regulatory T cells are impaired in HLH, and however, even though the Treg pool could be restored, the mice did not show a better survival. So Tregs alone were probably not strong enough to counter-regulate this overwhelming population of CD8 T cells. So the three major pathomechanisms that have been explored, which link deficient cytotoxicity with HLH, are first, a reduced clearance of the APC, leading to an increased synapse time with repetitive calcium release within and thus active TLs. And second, lack of fratricide killing. And third, an excessive consumption of IL-2 by CTLs, causing a relative lack of regulatory T cells. So, but now what about the antigen presenting cell? What about the other player in the immune system? So generally, dendritic cells play a critical role in driving T cell activation, and cytolytic destruction of dendritic cells by cytotoxic lymphocytes had been shown before. Therefore, one might think that there would be a quantifiable loss of dendritic cells in wild-type mice upon LCMV infection. However, as nicely shown by Michael Jordan and his research team, there was no difference in dendritic cells between wild-type and perforin-deficient mice. Therefore, the authors compared the function of the dendritic cell population from wild-type and perforin-deficient mice, and they nicely showed that the dendritic cell from the perforin-deficient mouse was a better stimulator and induced more interferon gamma production in T cells, while the wild titer at this time point of dendritic cell isolation was the same. And by addition of a caspase inhibitor, which inhibits apoptosis, wild-type dendritic cells also became potent stimulators, showing that the persistence of antigen-presenting cells is an important factor in the hyperactivation of T cells in this setting. So, in order to hitherto unknown genetic defects linked to immune dysregulation, we run the genetic study of immunodeficiency in my lab. We therefore recruit patients with unexplained immune dysregulation disease from our own hospital, from other clinics within our country, as the patient with the XLP1 that I showed you before. But we also recruit patients from abroad, as symbolized here by the Jeffrey pyramid, because we have a very nice collaboration with colleagues from Cairo. We then perform next generation sequencing with germline DNA, analyze the data by an in-house software, which we call the PID Butler. And if known PID is found, we combine the clinical and the results from transcriptomic analysis and try to come up with a hypothesis that could link the candidate gene with the patient. And then we have several nice tools which we can employ them. We have functional assays, and if necessary, we use animal models which to investigate the link between the genotype and the phenotype, and also, if possible, of course, to test targeted treatment in vitro or in preclinical models. And another patient whom we have included in our genetic study of immunodeficiency, was a now 25-year-old patient who has been suffering since the age of 9 months of recurrent self-limiting episodes of fever, splenomegaly, accompanied by rash, hemophagocytosis in the bone marrow, and elevated triglycerides. And as you can see here, as symbolized by the red dots, he had during episodes, he had blood count which returned to normal values, as symbolized by the yellow dots and the blue bars, symbolizing the reference values. And he also showed elevated HLA-DR, especially on CD8 T cells, but he had only lowish IL-2 soluble IL-2 receptor in his serum, while elevated ferritin. So he fulfilled the diagnostic criteria of HLH, but he had a peculiar cytokine pattern. He had lowish IL-2 receptor and interferon gamma, which are typically higher in patients with primary HLH, but he had higher IL-6 levels than the patient with primary episodes. So we therefore excluded the genetic causes for primary HLH and also for periodic fever syndromes in this patient, and performed whole exome sequencing with germline DNA from him and family members. And after exclusion of silent and low impact and common variants, we filtered with our in-house software for gene variants associated with immune system processes. And by doing so, we found a heterozygous mutation in CD48 at position 220. And this serine at this position is especially important in post-translational modification of the protein, because it is important for the attachment of the protein to the GPI anchor on the plasma membrane. According to proteins, a substitution of this serine by tyrosine would impact the surface expression of CD48, and this was consistent with our findings, showing a low surface expression of CD48 on patients' lymphocytes and monocytes. The patient also showed reduced soluble CD48 that did not increase as much as in healthy and HLH controls during inflammatory episodes, so strongly suggesting that the low CD48 expression on the surface of his cells was not due to an increased shedding of the CD48 from the surface. But what is CD48? So CD48 belongs to the SLAM family of proteins and interacts with high affinity with 2b4 and with lower affinity with CD2. And the interaction of 2b48 is known to be important for the 2b4 bearing NK cells in mice, and this interaction can have both inhibitory and activating effects on the NK cells. In humans, the precise mechanism is not so clear, although data point to a rather towards a stimulating effect of this interaction. However, we therefore analyzed the NK cell phenotype in our patient, and we observed that he had higher proportion of CD56 bright, CD94 high cells, which are classified as immature, and he had low amount of CD56 dim, CD94 low cells. So he had a low proportion of cells that are claimed to be more differentiated and better killers. This went along with expression of interferon gamma in NK cells, as shown on the right-hand side, upon stimulation with IL-12. Transcriptomic analysis of PHA-stimulated PBMCs from the patient revealed a greater overall level of cytokine mRNA expression, including the mRNA of IL-6, when compared to healthy control. It was very nice to see that the differentially expressed cytotoxicity-related genes belonged to the CD48-2b4 branch of interaction. So higher activity of LAT-mediated branch of 2b4 signaling was evidenced by the upregulation of several target genes such as TNF-alpha, interferon gamma, and perforin, and description of the EAT2, a negative regulator of SAP-mediated 2b4 pathway that controls NK cell activation, was also upregulated. We then evaluated the effect of the CD48 mutation on NK cell mediated cytotoxicity. And first, as you can see on the left-hand side at the upper part of the slide, we analyzed cytotoxicity using EBV-infected target cells from healthy donors and the patient, and incubated them in different effector target ratios with autologous NK cells. And thereby, we observed a lower killing of autologous target cells in the case of the patient's cell combination. Second, and this is shown on the right-hand side, on the upper part, we incubated healthy target cells with CD48 blocking antibodies and observed a lower of these antibodies when compared with control antibodies. And third, as shown in the lower part, we transfected a cell line, so hex cells, with wild type and mutated CD48 and observed reduced killing of the hex cells if transfected with the mutant form. To address the effect of reduced CD48 expression on a systemic inflammation, we infected CD48 mutated mice with a lymphocytic choriomeningitis virus, and we observed exaggerated inflammation with hyperferritinemia, IL-6 hypercytokemia, and lowish levels of soluble CD25, so mirroring the laboratory parameters observed for the patient with CD48 haploinsufficiency. So taken together, our patient showed an HLH-like phenotype, and we think that a possible pathomechanism for inflammation could be that the NK cell maturation is impaired and that there is an increased resistance of the target cells to be killed. So additionally to the three major pathomechanisms which have been explored that link deficient cytotoxicity with HLH, there is a fourth branch, namely the persistence of potent immunostimulatory APCs, as demonstrated by Michael Jordan and his team in the murine model, which is important in the pathogenesis of HLH, and it is in accordance with the occurrence of HLH in case of an increased resistance of the APCs to killing, as we have described thanks to the characterization of this patient with CD48 haploinsufficiency. So hemophagocytic lymphohistiocytosis and HLH-like disease with an array of different inborn errors of immunity. They are characterized by overwhelming activation of macrophages and a variable degree of activation and proliferation of cytotoxic lymphocytes. Activation and proliferation of cytotoxic lymphocytes is most prominent in those inborn errors which cause deficiency in granule-dependent cytotoxicity, while the pathomechanism in the case of the other diseases is less well understood. And the disease I'll show you now probably belongs to inborn error which is associated with HLH, but belongs among the inborn errors of immunity linked with insufficient control of viruses and also an insufficient control of kind production. From my perspective, it all started with a 3-month-old boy who was admitted to our clinic because of deterioration of the general condition after having had 3 days of fever. On admission, this patient showed acute respiratory distress, liver failure, and HLH-like features. The septic workup showed presence of HHV6 and a basic HLH investigations showed that there were no hints for primary HLH, although he fulfilled the criteria of HLH. And on day 3 after admission, this patient additionally developed thrombotic microangiopathy. And despite support by the ECMO and treatment with steroids and ganciclovir, the situation deteriorated and the patient, we had to withdraw day 6 after admission. And you can imagine how terrible it was when the mother called me 5 years later that the sister of the deceased boy suffered from fever with thrombopenia, and how desperate we were not being able to control the girl's inflammatory disease, who ultimately also died from a disease with HLH-like features. And she also had a central nervous disease, liver failure, thrombotic microangiopathy, and lung bleeding. We included the family in our genetic study of immunodeficiency and revealed a very rare, predictive damaging compound heterozygous mutations in both patients by whole exome sequencing with compatible family segregation. And collaboration, we were able to include 7 other families from all over the world in our study with a total of 15 patients, whereof genetic analysis had been done in 13 patients, revealing homozygous and compound heterozygous mutations in ZNFX1. The patients suffered from a severe disease with high mortality rate. Only 4 patients are currently alive, with 7 deaths occurring before the age of 3 months. The disease was characterized by virally triggered inflammatory episodes with tetanus involvement and early onset seizures and intracranial calcifications, lung disease, HLH-like disease, liver failure, and kidney disease. The severe reaction to viral infections were caused by both DNA and RNA viruses. It's noteworthy that measles and varicella zoster live virus vaccines caused severe vaccine strain infections in 2 patients. And although ongoing disease manifestations in some patients could be attributed to persistent and relapsing viral infections, other patients showed progressive disease even after the virus had been cleared.
All right. Oh, good. Yeah, you're back.
I'm so sorry.
We lost you again. So
yeah.
So all right. Well, hopefully.
I'm gonna finish in 4 minutes. I promise.
All right. Excellent. All right. Let's go for it. Thank you.
So we found these mutations, and they were spread all over the gene, and we made this model. And some of the missense mutations were, if you look in the 3D structure, close to the ATP binding site. So but however, yeah, it's not enough. We cannot draw any conclusions concerning genotype phenotype. But about ZNFX1. So it has been known from a mouse model that ZNF deficient mice are prone that ZNF binds to RNA viruses, and that the mice show a higher mortality if they are infected with an RNA virus than wild-type mice. And a previous publication has shown in C. elegans that ZNFX1 is important in evenly distributing small RNAs along long RNAs. And because of the patient's susceptibility to viral infections, Laura Faletti from Stefan Ehl's group infected monocytes from a patient and could show that even though when she stimulated by intracellular RNA, so kicking on the interferon stimulated, the patient's cells were less capable of getting rid of the virus than wild type cells. And I'll skip this. We thought about, okay, if they don't have the potential to get rid of viruses, what about the interferon stimulated genes? And to our surprise, the patient cells showed not a downregulation of interferon stimulated genes, but rather an upregulation, as shown in this transcriptomic analysis here. And if you focus on the middle figure, this is probably pathophysiologically the most important figure of the paper. We have seen that the mRNA of interferon stimulated genes has a longer half-life if in ZNF deficient, ZNFX1 deficient cells. So it seems as if the ZNFX1 responsible for degradation of certain RNAs, while the housekeeping RNAs or the degradation of the housekeeping RNAs is the same in wild type cells and ZNFX1 deficient cells. And we think, and this is also in accordance to a recently published paper, that the ZNFX1 plays a role in stress granules, namely these are membrane-less organelles, which are important in the degradation of mRNA. So probably in ZNFX1 deficiency, there is a problem in sensing the virus and also in regulating the half-life of certain mRNAs, especially the ones of interferon-stimulated genes. And then a big question mark in these patients is the treatment. Here you can see the MRI images of a patient who has been transplanted, so he has undergone hematopoietic stem cell, who had a severe ZNS affection before transplant and ameliorated after transplantation. And this goes along with the tissue expression of ZNFX1, which is more expressed in the hematopoietic system than in other tissues. But we still do not know how to treat these patients because we have observed two patients, patient 1.2, who has undergone lung transplant and did not suffer from any inflammatory episodes between the age of 10 and 15. And another patient who had recurrent inflammatory syndromes until the age of 4 years, but now is stable and is not suffering from any inflammatory episodes anymore. So this is a big question mark that remains for these patients. And with this, I would like to thank especially Stefano Vavasori, Laura Faletti, who performed most of the experiments. And I would also like to thank Stefan Ehl, Raif Jiha, and Tony Roschioli for a very stimulating collaborative atmosphere. The foundations for supporting our research, and of course you for your attention, and really sorry for my bad internet connection.
All right, excellent. Thank you, Jana, and thank you for staying on time despite the interruption. So very impressive, and very impressive work as well. So if you have questions, then you can use the raise hand feature in the chat so that we can identify you and unmute you and allow you to ask your question. I have a sort of two quick questions. One is that in every case, is it well, maybe the ones you just showed actually are the exceptions, right? But in most cases, perhaps, are you able to identify a virus that's been a trigger for at least an episode, if not every sort of manifestation? And then also, as these last couple of individuals suggested, this had sort of almost a chronic, not just acute episodes, but sort of almost a chronic aspect to it in some patients. And are those chronic episodes always kicked off by viruses as well?
So we have observed patients in whom no virus could be identified anymore and who continued to have especially the kidney manifestations, so this thrombotic microangiopathy, but also nephrotic syndrome and membranoproliferative glomerulonephritis, and also unexplained inflammatory episodes, which were not easily to be defined, whether it's vasculitis or just systemic inflammation, without finding a virus. Yeah.
Okay.
I don't understand why this inflammation should go on.
Right. And now, I don't. Let's see. I see one hand raised from Ed. Ed, you want to go ahead and let's see here. Ask your question. Yeah, you got it. Yeah.
So I apologize if I missed it. Was at least the two kids that you presented looked like they were very young at their initial age of presentation. Do you have a sense of the range of age for initial presentation with this?
Yeah, if I might eventually go back here. So they usually present young, and here. So the ticks represent patients who are alive, and the other ones died. So the median age of death was around 12 months. So not all of them had their episodes so early, but still some of them. And then a following paper by the Casanova group showed patients with mycobacterial infection. And they did have siblings who died at early age, but they were not clinically not described. So their phenotype was not described. So I think it might start early, but probably there are some patients who, for reasons we do not know, do not suffer from inflammatory episodes upon viral infections. So it is quite a heterogeneous phenotype, probably.
Thank you.
Okay, excellent. All right. Well, so if there are other questions, I say put them in the chat, please, because we need to move on to our next session. Thank you again, Jana, for that excellent talk. I had more questions, but of course, I don't have time to ask them, so we'll have to catch up later.